turning on your video feeds if you can for a few moments. I'd like to see who you are. Then you can turn it back again, turn it off again if you like. Okay, welcome back. Some of you are still coming on. Okay. All right. So welcome back. And so we are going to be looking at a very interesting topic today, Mycobacterium tuberculosis, as well as related organisms. Okay. So let me go straight to the presentation. Okay, you should be able to see the presentation now. So the topic today is Mycobacteria and actinomycetes. The bulk of the lecture is going to be devoted to Mycobacteria, of which there are two major species. One you know very well, Mycobacterium tuberculosis. The other is Mycobacterium leprae. Right? All right, so let's begin with the first slide, objectives. This is a slide that gives you an idea of what is important, what isn't important. So our task here in this lecture is to distinguish the features of Mycobacterium tuberculosis, um, and how that differs from other gram-positive and gram-negative organisms. Also, our objective is to summarize key features of the epidemiology, transmission, pathogenesis, and diagnosis, as well as treatment of tuberculosis, and then identify key features of Mycobacterium leprae, the organism that causes Hansen's disease, as well as the epidemiology, transmission, pathogenesis, diagnosis, and treatment of Mycobacterium leprae infections. Okay. So finally, we also will try to identify the distinguishing features of a number of bacteria that are called atypical mycobacteria that cause TB-like diseases, but only in immune-compromised individuals. Okay? Finally, we devote a small amount of time to actinomyces and nocardia, uh, two bacteria, two types of bacteria, which are grouped together with mycobacteria because of some shared morphological features. We'll come to that shortly. The bulk of the time is going to be devoted to Mycobacterium tuberculosis. Okay, so we will begin with reading a case history. This is normally a case history runs to 3 or 4 pages. Here it has been super compressed to fit in one single slide. I'm going to read this with you. Certain words and phrases are bold-faced. We're going to come back to this case at near the end of the TB lecture and revisit the same slide again. And this time we'll be better prepared to understand the significance of why those words and phrases are bold-faced. All right, let's begin reading this. The patient, by the way, this is a case, actual case I pulled from the literature in 1990s at some point, okay? But I compressed this to fit a single slide. The patient is a 37-year-old Asian female who originally presented to her primary care physician 2 years earlier complaining of cough. A PPD, or purified protein derivative test, was positive at 20 millimeters. A chest x-ray showed right upper lobe 3 centimeter nodule. The patient was told that TB should be ruled out. The patient's husband became angry and they went to see a pulmonologist. 2 years later, the patient returned to her primary care physician with the same cough, but this time she was coughing up blood. Arrangements were made to have the patient admitted to a hospital. The patient's husband became extremely upset the TB was mentioned again and stated that TB was impossible since he and his wife had received BCG vaccination. The patient and the family went into hiding. The social history of the patient. Patient was born in China, in the United States, almost 10 years with travel to China, volunteered at school, and was a homemaker with two kids. Husband was a professor at a major university. The case reported to health department and a court order was obtained after extensive and exhaustive efforts were made to have the patient be evaluated voluntarily. Sheriff's office was involved, but the patient could not be located. After patient finally gave a sputum sample in California to a sister who worked at a medical school, acid fast bacilli were found on a smear. Court ordered treatment followed in California. The husband had an abnormal chest x ray. Both children were infected without disease. So, this case is the framework, as we discussed Mycobacterium tuberculosis, is pathogenic pathogenesis, is treatment, and preventive measures. Okay, let's first get a perspective on tuberculosis. According to the World Health Organization, one third of all humans are infected by TB. This is a staggering number. This is unlike any other pathogen that we would have studied during the course of this course, right? About 10% of those 2 billion people will get sick, and some specific numbers are for 2015, more than 10 million people had active TB, and there were nearly 2 million deaths. Okay, these numbers are comparable to deaths from malaria, which is a disease of a long-standing origin and AIDS, and this disease far surpasses other recent diseases such as SARS. We're not talking about COVID-19, SARS and West Nile disease, West Nile virus disease, so on, both of which got a lot of publicity, but the numbers are very small relative to morbidity from TB. What I've inserted on the right hand side are numbers I pulled just today about COVID 19. Of course, COVID 19 is a pandemic, and before the disease settles in, we expect a vast number of infections and deaths as well. Naturally, here are the numbers: about 115 million people were infected. There are more than 2.5 million deaths, and so on and so forth. But these numbers, remember, are the numbers from a pandemic. Whereas TB is an example of a disease that's endemic, that stays in the human population. We'll discuss briefly where the organism is from and all that sort of information. But these numbers I've inserted on the right hand side on coronavirus um, basically tell you that bad as the coronavirus pandemic is, TB, the endemic disease TB, is nearly as bad, and it's always been with us and is likely to be with us for a long time to come. Okay. All right. So let's move on to the next slide. Look a little bit at the history of TB. TB, as we mentioned, is an ancient human plague, but it became really rampant in Europe, coinciding with rapid industrialization and urbanization. There were an astonishing 1 billion deaths attributed to TB between the in one century from 1850 to 1950. Okay? Now, at one time in the 19th century, before Koch isolated the organism, it was fashionable to believe that consumption, which are the name given to TB, was the disease of sensitive autistic individual. That's because many intellectuals and artists of that era, you'll recognize many of the names here, were afflicted by TB. And at that time, of course, people assumed that artists and intellectuals were sensitive individuals and who were prone to this disease called consumption. But of course, now we know that TB is actually an infectious disease, and the reason so many artists were affected was simply because artists tend to be poor. They live in crowded housing conditions, which make it possible for infection to spread widely. Mm, Okay, let's look at the epidemiology of tuberculosis. The reservoir for this organism is the human population. This strain of bacterium does not exist anywhere outside the human population. It cannot dwell in animals. It does not. There are related species in animals, but not this particular strain, and it does not live by itself in any ecological niche. No. So it's a strictly a human population reservoir. The current strain is relatively recent, um, in the sense that it probably around about 30,000 years ago. Okay? Now, MTB DNA has been found in Stone Age settlements in the Mediterranean region, and a disease similar to TB has been described in ancient India and in Greece, and features very similar to bone TB were observed in mummies from Egypt. Okay? We already mentioned that the disease reached epidemic proportion in 18th through 19th century, coincident with rapid urbanization and creation of crowded urban conditions. Right? We already mentioned that nearly a quarter of humans are infected. Maybe a quarter to a third of humans are infected, and the numbers are vast. The number of people who are infected, the number of people who have active TB, the number of deaths, all of those are big numbers. In the United States, the graph at the bottom right shows in the United States, there has been a gradual decline beginning at the start of the century until about 1980, into 1980s. Then there was a brief uptick coincident with HIV infection, but there has been a subsequent decline. This is where we are in recent years, approximately 10,000 to 15,000 new cases per year in the United States. But importantly, 4% of the American population, which is a little over 11 million people, have latent TB or LTBI. It's very important to understand that latent TB is not TB. We'll understand what latent TB is shortly, but as many as 11 million Americans have latent TB infection. Okay. We previously mentioned that the organism was isolated by Koch, um, Hermann Heinrich Robert Koch, in 1865. And it was also the time of the origin of the so-called Koch postulates on establishing the relationship between a particular organism and a disease. The famous Koch's postulates are still good today, more than a century later. And those postulates are summarized here. Of course, they had to be modified with new advances, but the foundation laid by these postulates is still quite strong. So, postulate one is that microorganisms must be found in abundance in all host organisms suffering from the disease, but should not be found in healthy organisms. Well, today we know, though, that there are exceptions. There are asymptomatic carriers. Okay? Right. Postulate two, the microorganism must be isolated from a diseased organism and grown in pure culture. Right? This is also mostly true, except for certain particular diseases, such as prion diseases. Okay? Postulate three, the cultured microorganism should cause disease when reintroduced into a healthy organism. Four, the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. So this is a rather rigorous set of rules, but obviously also very sensible set of rules, and these are still good today when you try to associate an organism with a disease, of course, with modification. Modification being asymptomatic carriers exist, and that there are certain diseases, such as prion diseases, there where you cannot culture the organism. Okay. All right, let's take a look now at mycobacterial morphology. First of all, Mycobacterium tuberculosis is a gram-positive slender rod, but it doesn't stain like a classic gram-positive organism. Okay, its staining is actually ambiguous with gram stain. This organism is non-motile and is non-spore forming. The fundamental, a fundamental feature of Mycobacterium tuberculosis morphology, and one that you must get to know well, is that the cell wall is very peculiar. The cell wall is extremely lipid rich. A major cell wall lipid is called mycolic acid. There are, however, other lipids as well. The cell wall also has certain characteristic um, lipid carbohydrate molecules such as lipoarabinomannan and other components of peptidoglycans such as N-glycolyl muramic acid, etc. The fatty nature of the cell wall um, creates particular conditions that have been exploited in identifying the presence of this organism in a biological sample. And this staining feature is called acid-fast staining. We're going to learn about that in another slide that's upcoming. But first, more information on the cell wall structure with a more detailed drawing on the right-hand side that tells you what the cell wall of a Mycobacterium tuberculosis bacterium is. So, starting with the plasma membrane or cytoplasmic membrane, which is a lipid bilayer, um, there is a thick peptidoglycan layer, but they're topped with mycolic acid, which is the red wiggly lines throughout. Okay, so the entire surface is extremely lipid rich as a result of the presence of this fatty acid as well as other fatty acids that are present at the surface. Now, this creates an extremely hydrophobic cell surface. Now, in order to survive, the organism has to be able to get nutrients, water, and other essentials back inside the cell. Therefore, there needs to be a mechanism for uptake of small molecules and water. And that provision is made by inserting proteins called porins at the cell surface, which are basically channels that allow for uptake of hydrophilic molecules and deliver them into the cell interior. As we previously mentioned, the principal contributor of hydrophobicity is mycolic acid, which is anchored to a glycan such as arabinogalactone. This is the arabinogalactone entity. All right. In addition to this, you might be wondering if the antigenicity, if any, of Mycobacterium tuberculosis is to any of the surface molecules, which is the norm for many bacteria. It turns out that for Mycobacterium tuberculosis, the major antigenic component is not a surface molecule at all. These are molecules actually called liberated proteins, and they're given the collective name tuberculin. Tuberculin is not a single protein, it's a mix of dozens of different kinds of proteins. which are only released by the bacterium upon death. Typically, tuberculin is prepared by autoclaving a culture of Mycobacterium tuberculosis and then removing the debris and taking the soluble protein fraction. Okay? So the major antigenic component is not a surface molecule, but rather a liberated protein that are only liberated from dead cells. Okay. Here we come to the peculiar staining property that is used for diagnostic purposes and until recently is the main diagnostic procedure for initially identifying infection with Mycobacterium tuberculosis going back to more than a century. The acid-fast staining procedure is described here. It's also given the name Zeal-Nielsen or ZN staining procedure. It depends on the following features of mycobacteria. In the first step, you stain a biological sample. Any biological sample that you suspect may have Mycobacterium tuberculosis is also likely to have other normal commensal bacteria as well as, of course, host cells, etc. So you stain a biological sample with a dye called carbol fuchsin, which is a pink dye. Basically, the pink dye is taken up by all bacteria and also by all host cells. Okay, so everything stains pink in this step. In the next step, you take the dried smear uh, and then you decolorize it by washing the smear with acid alcohol. When you put acid alcohol on a dried smear of bacteria from biological sample, then most bacteria that are not acid fast will give up the color and become completely decolorized. That is, all host cells as well as all other bacteria will give up the color and become totally colorless. The organism that is acid fast, such as Mycobacterium tuberculosis, will not give up the pink color. It remains pink. However, to visualize it more effectively, in the next step, you counter stain with another dye, a blue dye called methylene blue. When you counter stain with methylene blue, all those cells, bacterial or otherwise, that became colorless will take up methylene blue and become stained dark blue, thereby contrasting with the pink acid fast Mycobacterium tuberculosis cells. So what is shown here on the right hand side upper part is a sputum sample, which is typically the sample that's analyzed for a TB patient with active TB, where you see pink rods which represent mycobacterial species. This staining procedure, by the way, I should tell you right away, is not specific to TB. Any mycobacterial species will stain that way. Okay? So sometimes without TB, you could see occasionally a pink rod, but in TB patients, you'll see an abundance of such organisms. What is shown at the bottom is a lab mix for the purpose of demonstrating acid-fast staining. In this case, two different kind of bacteria have been mixed together. One is acid-fast, the other is not acid-fast. And at the end of the Zeal-Nielsen staining procedure, you see some cells are pink, the others are blue. Okay. All right. In this cell, we consider certain growth characteristics of Mycobacterium tuberculosis that are actually relevant to its pathogenesis. The number one fact about the physiology of Mycobacterium tuberculosis you should know about is that it's a strictly aerobic organism. That makes sort of sense because it's a disease that affects primarily lungs, right? It's a strictly aerobic organism. And the second most important thing you need to know is that it's an intracellular organism. It grows only inside a host cell. It cannot replicate outside a host cell. uh, So, in this case, they reside and proliferate within host macrophages. This organism is quite resistant to drying and to disinfectants. All of that is attributed to the thick lipid-rich cell wall, but it is sensitive to heat. Okay, in fact, pasteurization was invented mostly to get rid of mycobacterial species in contaminating milk and other such dairy products. This organism can grow in vitro at 37 degrees, but it cannot grow at room temperature. So it is adapted to growth at 37 degrees because its reservoir is human beings. Okay? However, to grow it in vitro, you need an extremely rich, very complex medium, such as the Lowenstein Jensen medium. This medium is essentially based on egg yolks. So, unlike the kind of medium, simple medium that you can use to grow normal lab bacteria like E. coli, you need a very rich, very complex medium of the kind that's provided from biological material, such as egg yolks. The organism grows extremely slowly, dividing only once about a day, 12 to 24 hour cell division time. And colony formation naturally takes about a month. Okay, and the colonies have a characteristic appearance. They have clumped appearance. The clumped appearance is because of the fatty nature of the cells. Okay, Mycobacterium tuberculosis infection is spread by droplet nuclei. You are all now completely, totally familiar with the droplet nuclei, and thanks to the pandemic, you know all about it, right? So it turns out that tuberculosis is spread through the same mechanism. Droplets which are expelled, invisible droplets that are expelled when a person who has active TB, this is very important to understand, only person with active TB, not any person who has latent TB, but only active TB. When such a person coughs, sneezes, speaks, or sings, that person is emitting droplet nuclei. In this image, there is a dramatization of the of droplet nuclei, but in reality, droplet nuclei will be invisible to you. You just inhale them when somebody in the room is emitting them. Mm. Close contacts are at the highest risk of becoming infected, and transmission occurs from person with active infection only, not from patients with latent TB infection. That's very important to understand. Okay. In this slide, this is first of several slides devoted to the same topic. We get an overview of the consequences of exposure to Mycobacterium tuberculosis. On the left-hand side is a chart that tells you what happens once you inhale droplet nuclei that contain Mycobacterium tuberculosis. Incidentally, this is an extremely infectious agent. You don't need very many bacteria to become infected. It may be as few as one or two, or maybe a few dozen. It's not a huge number. Right? All right. The initial exposure and inhalation of a droplet nuclei would lead to uptake of the organism by alveolar macrophages, those are macrophages in your lung, and limited initial intracellular multiplication, followed by formation of something called granuloma. We'll come to a granuloma, which is shown on the right-hand side eventually, but for now, just stay focused on this chart. So initially, a limited amount of intracellular multiplication within macrophages lead to granuloma formation. After the granulomas are formed, there are two possible fates. For a majority of individuals, in fact, 95% of the individuals who are exposed to TB infection through this route, they become asymptomatic. Okay? Here, in this case, the bacterial multiplication stops. The bacteria do not quite die, but they remain contained indefinitely. And the contained bacteria are within structures called tubercles, and they can sometimes become calcified. They can remain so for the entire lifetime of many of those individuals. But among that 95%, 5% can suffer reactivation TB, but the remaining 90%, which is the vast majority, will remain asymptomatic throughout their lifetimes. Okay. What about the 5% who immediately after the initial exposure to Mycobacterium tuberculosis, what about these 5% who go into primary TB? What happens in those individuals? What happens is, in these cases, bacterial multiplication continues. Bacterial multiplication leads to spread to other locations within the lung, eventually leading to clinical symptoms um, that are provoked by tissue damage. And this is the condition called primary tuberculosis. We'll look later at what happens in the lungs of primary tuberculosis, what the symptoms are. Similarly, in reactivation TB, which occurs at some point in the lifetime of those 95% who did not show immediate disease, reactivation TB again is characterized by bacterial multiplication, spread, and tissue damage. But typically, reactivation TB tends to have more serious manifestations than primary tuberculosis. A question you might want to ask right here is, what determines this 5% who become um, uh, tuberculous immediately after infection, and why are this 5% suffering reactivation? Well, we don't know answer to those questions, but the underlying presumption is the fraction that gets the disease or those that are somehow compromised in their immune responsiveness, either lack of immune response or an overreaction of the immune system. Okay, we'll come to those topics in later slides. But here, let's take a look at a granuloma. What is it? What does it consist of? What is shown at the bottom is a micrograph of a granuloma, is the structure, and what is shown above is a cartoon version of a granuloma. The granuloma is formed not only in response to Mycobacterium tuberculosis, but for several different organisms, many of which are actually related to Mycobacterium tuberculosis. These are called these cause diseases called granulomatous diseases. So what's the granuloma? In a granuloma, granuloma is an area where infected macrophages are walled off in a physical small fortress like structure, which consists of mostly immune cells. For example, in this cartoon depiction of a granuloma, there's an outer ring of green cells. These are these are lymphocytes. In the interior of the granuloma are macrophages. Macrophages are these orange shape, some of which are infected. Those are the dark oblongs. So some macrophages are infected and have TB, bacilli inside them, but other macrophages do not have the bacilli, but they're also part of the granuloma. In addition to this, there are several other kinds of immune cells here as well, such as foamy macrophages and so on and so forth. But the important point here is, I don't need you to memorize the name of each type of immune cells here. All we need to know is that granuloma is a walled off area consisting of immune cells, embedded within which are macrophages which carry TB bacilli inside them. Okay, so Ultimately, the granuloma will be enveloped by a fibrous cuff, shown here in cartoon form as wiggly blue lines. So this is like a physical barrier is created around the granuloma, and this granuloma in an infected person has several possible fates. In case of those individuals where there is there is a progression to primary tuberculosis, certain events happen within the granuloma. And in those patients where exposure has occurred, but those who have become latently infected without any sign of primary tuberculosis, the granuloma undergoes further changes. Then, upon reactivation to reactivation TB, there are other changes in those granulomas as well. We'll go through each of these varieties of changes in subsequent slides. So, for here, the important part is to understand what a granuloma is. Granuloma can be viewed as an effort by the host organism to isolate infected macrophages in a fortress like structure consisting mostly of immune cells, which are surrounded ultimately by a fibrous cuff. Eh? All right. So here is another view of a granuloma and its relation to other structures called a tubercle and and a caseation focus. Excuse me. This is one of a series of slides I'll be showing you throughout this lecture. So, when a Mycobacterium tuberculosis, the blue oval here, is taken up by an alveolar macrophage, that the macrophage becomes surrounded by immune cells to create the granuloma. The granuloma matures and is encased in a fibrous cuff. Okay. Then, in late stages, when reactivation occurs, the something called caseation or liquefaction of the interior of the granuloma occurs, and eventually, if the circumstances allow it, the granuloma bursts open, releases bacteria into the environment where they can be taken up by other macrophages and rapidly multiply and amplify their numbers. Okay. So, what forms initially is the granuloma, and in the and later granuloma with a fibrous cuff. What's a tubercle? A tubercle is a mature granuloma which has further which has further been fortified by calcium deposits so that it becomes radio opaque. Radio opaque means it can be seen as a uh, from X-ray scattering on an upper thoracic chest X-ray. So a tubercle shows up as a small nodule that is that that blocks X-ray, so that it casts a shadow in the chest X-ray. That's a tubercle. The tubercle then is a calcified version of a granuloma. A caseation focus is a granuloma where liquefaction of the interior with eventual release of bacteria can occur. All right, that let's move on to the pathogenic mechanisms of tuberculosis. All right, from a previous lecture by me, as well as another lecture on pathogenic mechanisms by another instructor, you'll learn that many bacteria kill the host cell through powerful toxins. But that's not the case for Mycobacterium tuberculosis. Mycobacterium tuberculosis, which is a deadly bacterium, nevertheless makes no exotoxins. It has no overt virulence factors either. Okay. So its ability to kill the host resides in its ability to survive and multiply within macrophages. Now, you may recall from other instructors' lectures that macrophages are an extremely hostile environment for a bacterium. However, Mycobacterium tuberculosis has evolved mechanisms to survive the hostile environment of a macrophage. Now, Mycobacterium tuberculosis elicits protective immunity. It can elicit protective immunity, that is adaptive immunity, but, but it also provokes pathological hypersensitivity that ultimately actually what leads to tissue damage that is characteristic of TB. That is, MTB kills by turning the immune system against the host. All right. This slide is devoted to trying to understand how NTB can survive within a macrophage. It uses multiple mechanisms, but one major mechanism is shown here, and that is MPB has the ability to block a physiological process within macrophages called phagosome lysosome fusion. Okay, here is what this phrase means. In a macrophage, shown here in the large wiggly diagram here, uh, normally the job of macrophage, among many other jobs, is to pick up bacteria and to internalize them through phagocytosis. The internalized bacterium is inside a compartment called a phagosome. Now, the purpose of taking up bacteria into the interior, into phagosomes, is to eventually target them to killing. A major mechanism for targeting them for killing is to fuse the phagosome with another intracellular organelle called a lysosome. Lysosomes are basically internal organelles that are full of degradative enzymes, enzymes that can destroy bacteria. Okay? Because they can also destroy the host cell itself, they are contained within vesicles called lysosomes. So one of the things that happens to a phagosome is that it gets fused with lysosome, and this process creates the so-called phagolysosomes. In a phagolysosome, the enzymes that are present in the lysosome act to destroy the bacterium. That is a defensive mechanism against bacteria. So MTB blocks this process. This fusion is blocked by MTB. As a result of that, the bacterium, even though it's contained within a phagosome, is not killed. This is believed to be a major mechanism by which MTB survives and ultimately leads to disease. Okay, we already noted that only about 10% of infected humans ultimately develop disease. 5% immediately, another 5% later on, some stage in their life. Okay, this says that there is a high level of natural immunity. Okay, having said that, we do know that certain population groups are more at risk for disease. That is, the number is not 10% for everyone. It is 10% for normal humans, but the percentage is higher for people with certain underlying conditions. Okay. Now, upon exposure to MTB, cell-mediated immunity, sometimes I'll refer to it as CMI, develops within 2 to 6 weeks after primary infection. And as we previously mentioned, what causes the disease and ultimately death is a perverted immune reaction called delayed type hypersensitivity to antigens, MTB antigens, that really determines disease outcome. Okay. Mm. In this chart here, we will undertake an overview of TB immunopathology. Okay. My purpose here is to not to go through a lot of immunology, but just at an overview level. This and another slide. Okay. So upon ingestion of MTB and uptake by macrophage and other immune cells, there is limited multiplication of MTB, but that leads to secretion by the infected macrophage of certain cytokines and chemokines. Now, cytokines are small protein molecule, signaling molecule that act on other cells, including other immune cells. Okay. Now, chemokines are a kind of cytokines which have the purpose of recruiting other immune cells. They're sort of chemo attractants for other immune cells. Okay, so all right. So infected macrophage produce cytokines and chemokines, which collectively lead to T cell activation, which in turn lead to further production of cytokines and formation of cytotoxic lymphocytes. Collectively, collectively, these actions lead to granuloma formation, and a limitation on the amount of multiplication that's possible. However, in certain individuals, that is about 5% of the population, this attempt to limit the multiplication is not successful, leading to primary tuberculosis. But for the vast majority of people, 95% of the people, the intervention by the chemokines and the cytokines is sufficient to lead to successful granuloma formation and containment so that these individuals can remain asymptomatic virtually for an entire lifetime, except for that small percentage which at some random time in their lifetime can suffer reactivation TB. When either reactivate when MTB is allowed to multiply again, either as a result of primary TB or reactivation TB, ultimately there is delayed type hypersensitivity mediated tissue destruction, and that leads to active so-called caseating necrosis and bacterial release and extensive tissue damage, cough, and expulsion of droplet nuclei containing MTB. So restarting the cycle. Okay, this is an overview of the immunopathology. Some details are shown in this and this slide. Okay, okay. So this slide has basically nothing new from other than what I have mentioned in the previous summary slide, but just for the purpose of um, um, no, for the purpose of fortification of the what I told you. Here are the important part. Oh, by the way, very important. You don't need to memorize the names of individual cytokines or chemokines here. There are only about two or three names that I'll mention that are actually important that you might want to remember. So an infected macrophage, shown here in diagrammatic form, produces a number of cytokines and chemokines. Okay? An important cytokine whose name you might want to remember is TNF alpha, tumor necrosis factor alpha. Okay? Which and the chemokines that are produced have the collective effect of recruiting neutrophils, which is a form of immune cell, and and these immune cells again produce more TNF alpha as well as other cytokines, and TNF alpha can also act on the same infected macrophage to further amplify TNF alpha, other cytokines, chemokines, and so on. Ultimately, these cytokines also will activate macrophage to create activated macrophage as well as trigger other immune responses. Okay? All right. This is another view of the progression of a granuloma or a mature granuloma called a tubercle to the stage of caseation. Okay? Now we mentioned that we mentioned that uh, in latently infected patients, the granulomas remain stable virtually the entire lifetime. Okay, but at some point they get reactivated. What triggers them to be reactivated is not known, but it is assumed to be some failure of cell mediated, some transient failure of cell mediated immunity. Okay? So, whatever the trigger is, the first step is that the macrophage die off and the center of the granuloma becomes liquefied. And this is a phenomenon that's called caseation because it is resemblance to cheese-like mass. Okay? Caseation. Okay? Once caseation forms, eventually the granuloma falls apart and the MTB are released. When they're released in the lung, they're exposed to oxygen. Oxygen acts as a signal for reactivating what was formerly dormant Mycobacterium tuberculosis and prepares them for multiplication. But remember that they cannot multiply in an extracellular environment. They need to be taken up by new macrophages, and they can only multiply inside new macrophages. Okay. This is a view of a gross pathology view of caseous necrosis, which is a major characteristic of TB pathology. What is shown in this segment of the lung, are areas which are, in this case, there are a very large number, hundreds of caseation foci. Caseation referred to cheese-like masses. Each of these masses is an area of tissue destruction, which is completely filled by dead immune cells as well as dead host cells. Yeah? Hmm. What is shown on the right-hand side bottom is a as an X-ray film showing extensive areas of radio opacity as well as the presence of the so-called cavities. Right? Cavities form in a lung when a site of caseous necrosis ultimately is liquefied and the liquid is expelled by coughing and leaving cavities that are filled by liquid. Okay, they show up on the X-ray as shadows as well. Okay, in a small fraction of patients, um, pulmonary tuberculosis can spread throughout the body to different tissues. And when that happens, that kind of tuberculosis is called miliary tuberculosis because of the presence of a large number of small tuberculous lesions scattered throughout different tissues in the host. Okay? This is a highly disseminated form of TB. It can occur in lungs, spleen, liver, kidneys, bones, brain, and so on. Okay? The mechanism of miliary tuberculosis is believed to be um, blood vessel erosion by a caseating necrosis in lung, which releases bacteria into the bloodstream or the lymphatic stream through which they can spread to other parts of the body and where they can form tuberculous lesion. Now, this miliary tuberculosis is rare in normal TB patients. Only about 1 to 2% of the immune competent people get miliary tuberculosis, but it's much more common in immune compromised patients, such as HIV infected patients. Okay? Miliary tuberculosis doesn't have a very strong clinical presentation, but it is 100% lethal if it is not detected and treated. Okay. Let's move on to clinical presentation of TB. What do you actually see in the clinic? Okay. Primary tuberculosis has mild symptoms, and the clinical presentation is fever and malaise. And a characteristic of primary TB is the presence of the so-called Gown's focus and Gown's complex. Okay. So what is Gown's focus and what is Gown's complex? Okay, Gown's focus refers to an X-ray shadow indicating a small calcified area or a tubercle. We know what a tubercle is by now. Okay, the small area that is shown, that's called a Gown's focus. Okay? A Gown's complex is the presence of such a Gown's focus in combination with swollen lymph nodes. Okay? That's called lymphadenopathy. Okay? Together it is called Gown's complex. This is a characteristic of primary tuberculosis. This feature, the Gown's focus and Gown's complex, is quite commonly observed in areas of the globe where TB is highly prevalent, such as India is one country. There are many other countries in the world where this is a primary diagnostic. There is Gown's focus and Gown's complex. Okay. Reactivation TB has actually very severe presentation, and this is characterized by prolonged cough and bloody sputum, hemoptysis, and chest pain. And they have high fevers, night sweats, malaise, extreme fatigue, and very severe weight loss. It's not uncommon for these patients to lose as much as 20% of their body weight within a matter of months. Okay? So radiography will show consolidation as well as cavities. Okay? And TB, of course, reactivation TB is lethal within 2 to 5 years if it's left untreated. Okay, now we want to ask, why exactly is TB fatal? Right? We already know that MTB does not make any toxin, does not make any cytotoxic agents. Okay? All it does is its uncontrolled growth causes destruction of lung tissue, largely not because of anything it produces, but because of the host inflammatory response. Right? So the lung damage, of course, ultimately uh, um, uh, damages the ability to breathe, to respire. Okay, so ultimately these patients die of suffocation. Okay? And of course, the sites, the cavities that are filled with water, in effect, make these patients drown, death by drowning, suffocation. That's why TB patients die. Okay? That's also the reason, by the way, liquefaction of the tissue, and the attempt of the body to expel that content via coughing is what allows the organism to spread from one host to the next host. Okay? Less commonly, death is caused because of infection and organ failure. This is miliary tuberculosis in other organs. Okay. We previously mentioned that when initially exposed, 10%, only 10% of people who are initially exposed to MTB ultimately suffer active disease. 90% never show any symptoms for the rest of their lives, right? Is that true for all populations? No, of course not. To no one's surprise, smoking dramatically increases TB reactivation and mortality. This is something that you would, of course, guess, but it still had to be formally proven. And a study that definitely proved that was conducted, a large study in 2003 in India. And this study proved that smoking, either cigarettes or BDs, which are native cigarettes, are linked to increased incidence of reactivation TB to the tune of 400%. Death is 4 times more likely for smokers with TB as compared to non-smokers in TB. Similarly, underlying metabolic conditions such as diabetes increase susceptibility to TB. And finally, also not surprisingly, Um, HIV infected patients are very susceptible to TB. That's because HIV disables CD4 T cells, CD4 plus T cell, which are required to activate macrophages and CD8 plus T cells that control TB infection. So HIV basically compromises the immune system and therefore enables TB to spread much quicker in an uncontrolled fashion. Okay? The incidence is dramatically increased, up to 100 times more frequently than compared to normal individuals. Okay? It also tremendously increases the risk of reactivating TB. So a latently infected individual, when he or she acquires HIV, is at tremendous risk of suffering reactivation TB as well. Okay? All right. How do you diagnose TB? Diagnosing TB today, the initial diagnosis is made by checking the response to the so-called tuberculin test. What is tuberculin test? Tuberculin test basically measures delayed type hypersensitivity to a challenge by a protein preparation prepared from MTB cultures. Okay? The protein is given various names, tuberculoprotein or PPD or purified protein derivative, and tuberculin, etc., etc. Basically, a certain standardized amount is injected into the forearm, and observation is made 2 to 3 days later, looking for an area of induration. Area of induration means a hard elevated area at the site of the injection. I'll show you a better picture of what that means in a subsequent slide. But basically, if the area of induration is 10 millimeters or larger, it is considered to be a positive test. Okay? Very important thing to note is that there are false positives, there are false negatives. Okay? False positive, a very prominent source of false positives is BCG vaccination people. That is pretty much everybody else who was not born in the United States is likely to be BCG vaccinated. Okay, so you have a large pool of false positives possible. Okay, and also occasionally, rarely, you can be infected by non-TB mycobacteria, in which case also you can be a false positive. There are also false negatives. For example, a condition called anergy, that is a failure of the immune response altogether, that is observed in immune compromised patients. For example, HIV infected patients will respond with a false negative, even though they might have active TB, okay? Because you don't see any DTH that gives rise to that area of induration. Okay, all right. So lab diagnosis is classically the lab diagnosis is based on acid-fast bacilli found in sputum or spears. Okay, so here is an example of a sputum sample from a patient that shows that there are acid-fast bacilli. However, we mentioned before that there are many mycobacterial species which do not cause TB disease, which are environmental in nature, right? So it is necessary for culture confirmation to make sure that it is Mycobacterium tuberculosis and also to determine whether there is any antibiotic resistance so that you can come up with a regime for treatment. Okay. Now, these are the classical methods, right? However, not surprisingly, today diagnosis can be made much more rapidly. These laboratory, the classic diagnostic tests, which are still practiced in many parts of the world, are, however, not only laborious, but they are very time consuming. Culturing and identifying antibiotic resistance can take a month or more, whereas PCR-based modern methods will take only a few hours to determine both the presence of Mycobacterium tuberculosis as well as any drug resistance that may be present by simply scanning for common mutations that confer drug resistance. Okay. As promised, this is a picture that shows what is meant by an area of induration. In this case, this patient has reacted with this induration here, this area of elevated skin. That's induration. The red area is not what is measured. It is this area in the center that is measured. When that is 10 millimeters or larger, then the test is positive. If not, then it's considered to be negative. Okay. So this is a cartoon telling you about a PCR test provided by the person who manufactures this test. The most important part of this slide is the part that is circled at the bottom, and that is the diagnosis is complete in less than 2 hours from a patient sample to determining whether it's Mycobacterium tuberculosis and also determining whether it is resistant to rifampicin. There are variations of the test that have been approved for the diagnostic laboratory, but how widely they have been adapted, I'm not entirely certain. Okay, now we return to the case history again, and after we reread the case history, we'll take a 5 minute break. Then we come back to wrap up a few additional parts of TB, and then we move on to other organisms. Okay, returning to the case history, I'm going to read this more rapidly, but I'm going to focus on the bold-faced letters. Okay, so our patient initially came in with a cough. You know why TB patients cough? They cough because they have suffered caseating lesions, which are getting liquefied, and the lungs are trying to get rid of the material, expel the material by coughing. Okay, a PPD, a purified protein derivative. This is the antigen prepared by boiling or autoclaving mycobacterial cultures, and then taking the soluble component to use as an antigen challenge, okay, to diagnose people who have been exposed to Mycobacterium tuberculosis. Positive 20 millimeter is significant because the definition of a positive test is 10 millimeters, and this patient had an area of induration that 20 millimeter that is well above the 10 millimeter threshold. Therefore, she definitely infected with Mycobacterium tuberculosis. A chest X-ray is a common diagnostic procedure because because here you can detect any cavities and any tubercles that are formed as a result of exposure to Mycobacterium tuberculosis. And the right upper lobe is significant because as an aerobic organism, the infection typically starts in the most aerated part of the lung, which is the top part of the lung. Okay, so right upper lobe is significant because of that. Okay, even though of course tuberculous lesions can form throughout the lung, they start in the upper lobe. Okay? So the nodule is significant because this is what that cat shadow. This is probably a tubercle. Okay? Coughing up blood is highlighted to show you that there is significant tissue destruction, including blood vessel erosion, that is releasing blood into the cavity that being coughed up, so that you have cough where sputum is tinged with blood. Okay? All right. The patient is upset. The patient believes that she cannot get TB because she was BCG vaccinated. Is that true? Well, we'll come to that when we return to the class after the break. Okay? But BCG is a vaccination given to children throughout the world, but not in the United States, for reasons we'll discuss soon. Okay? The reason we highlighted volunteers at school is because she has inadvertently exposed other individuals in a small confined area, such as a classroom. And homemaker with two kids is highlighted because she is, of course, living at home and exposing family members when she's coughing or talking or singing or whatever. Okay? And the case reported to health department is highlighted because TB is a prime example of a reportable disease. So if you have a patient who is identified, suspect has TB, you must report it to the health department. Right? And sputum is highlighted because that's the one of the primary diagnostics was is by collecting sputum samples and staining them for acid-fast bacilli. You already know what acid-fast bacillus means. Okay, we'll take a 5-minute break now, and when we return, we'll wrap up a few additional topics on TB before moving on to other organisms. 5 minutes from now, that will be 3:20. We'll be coming back. Good afternoon and welcome back. Okay, to wrap up a few additional topics on TB, let's first look at TB treatment and and drug resistance in bacteria, Mycobacterium tuberculosis. The key to TB treatment is that it's a long-term treatment. So for many bacterial pathogens, the treatment regime is consists of days and maybe a few weeks. But for TB, the treatment regime is 6 months to 2 years. It's a very long time. The second important thing to remember is that TB treatment is almost always with a combination of drugs. That is in order to prevent resistance to resistance to antibiotics. Okay, so because of these features, there are certain problems in treating patients, and the most important of them is patient compliance. It's very hard to get patients to take medication, especially when they're not feeling particularly sick for a very long time, like 6 months to 2 years, right? And it is exactly that situation that leads to creation of emergence of multi-drug resistant bacterial strains. Okay, so TB treatment uh, can be in the form of giving first line drugs or second line drugs in case the first line drug failed to control the disease. Okay, the first line drug example is not necessary from the perspective of my lecture to remember the names of all of these antibiotics and what they do. You might, however, have received this information from a dedicated lectures on antibiotics, in which case you're still responsible for learning that part. But only certain conceptual parts are important from my lecture perspective. Okay, many of the first line drugs are drugs that target cell wall. For example, isoniazid, ethambutol, both of these target cell wall synthesis. Okay, pyrazinamide target fatty acid synthesis, which again is related to the cell wall, right? So the only one member of the first line drug that's not something that specifically targets cell wall is rifampicin, rifampin is spelled either way, rifampicin or rifampin. This targets RNA synthesis only in bacteria, RNA synthesis in bacteria, right? So these are all first line drugs. Second line drugs are given in cases where first line drugs fail to control the bacterial multiplication. Second line drugs are drugs like para-aminosalicylic acid, ethianamide, cycloserine, fluoroquinolone, other uh, um, aminoglycosides such as amic acid, etc., etc. Okay? There are certain new drugs that have been recently approved, and those are two of them are beta-quilene and delaminate. I'm sorry, yeah, delaminate. Okay. Okay. It's very important to recognize that there are multi-drug resistant or MDR TB bacilli that are increasingly being encountered, and there are XDR for extensively drug resistant TB as well. Okay, so MDR and XDR have rather precise definitions, and those are summarized in this slide and also shown diagrammatically at the bottom. Okay? So MDR is defined as bacilli that are resistant to two of the best first line drugs, and those two are isoniazid and rifampin. So MDR-TB is when you have resistance to isoniazid and rifampin. XDR, on the other hand, is when an organism has not only resistance to isoniazid and rifampin, but it also has resistance to a fluoroquinolone plus one plus at least one of the so-called injectable second line drugs. So MDR, we're talking about resistance to two. XDR, we're talking about resistance to four. In MDR, the two are fixed, which is isoniazid and rifampin. In XDR, the first two are fixed, which is the same as isoniazid and rifampin. And the third is a class, fluoroquinolone. There are many different kinds of fluoroquinolones. All of them block replication in bacteria, plus one of three different injectable second line drugs. So that's the definition of XDR. All right. This map gives you an idea of the prevalence of MDR TB throughout the world. As you can see, the distribution is quite extensive. Right? It's a major area for MDR TB is Russia and Eastern Europe, but there are pockets present elsewhere as well. Okay. So how do multi-drug resistant mycobacteria emerge? Well, it's a process in which, because patients have been treated for a very long time, there are plenty of opportunities for multiple drug resistance to arise. So, so the process works something like this, as shown by this simple diagram. Okay, you begin with a patient who is infected by TB bacillus that has no resistance to any antibiotic. In other words, is sensitive to all antibiotics that work on TB bacillus, right? Now, a patient who is infected by TB has a large number of TB bacilli, right? Within any large bacterial population, there will be a small minority at the rate of like 1 in 1 million cells, which will spontaneously develop resistance to, for example, isoniazid or rifampicin and so on. So these are not very large fractions, one cell in a million. And this is a random process. Mutation, as you know, is a random process. Now, if this patient is treated with a drug that is quite effective in killing bacteria, such as isoniazid, right? This kind of treatment is called a monotherapy. If you give this patient isoniazid, it will undoubtedly kill most bacteria, 1 million bacteria, but that 1 million bacterium that is resistant to isoniazid, that will not get killed. And that will now expand into form a population of cells that are all isoniazid resistant. And if you subject this patient thinking that now you can attack it by giving it rifampicin, yeah, rifampicin will kill most of the isoniazid cells. However, 1 in a million of those would have developed also resistant to rifampicin, and those labeled here IR, isoniazid rifampicin, those will expand, so you have multi-drug resistant bacterium. So this kind of emergence of multi-drug resistance is very common and is often the result of so-called monotherapy. You can't just give one drug. That's the reason why you never treat TB with just one drug initially. Okay. Okay. So this is also about acquisition and transmission of MDR TB. There's nothing new in this slide. I'm going to skip it and go to the next slide. These are the two new drugs approved by World Health Organization for treating MDR TB. The two new drugs are bedaquiline and delamanid. Right? And the mechanism of action of bedaquiline is it blocks ATP synthase and mycobacteria. And it's bactericidal, it's quite effective, but it does have some serious side effects, cardiac side effects. So it has toxicity, but it is quite effective in treating MDR, MTB. Delamanid has a different mechanism of action. It inhibits cell wall synthesis, mycolic acid synthesis. It's also quite effective, but it also has side effects. Okay? But both of them have been approved for treatment of MDR TB. Okay. So, like with any disease, prevention is the best practice. In TB prevention, public health measures are basically infection control, isolating patients, that is, PPD test to monitor exposure to TB, X-ray, the part of the monitoring, maintaining case registries, contact tracing. You heard about contact tracing a lot in this pandemic era. And importantly, a public health measure is something called directly observed treatment. Directly observed treatment refers to the need to ensure patient compliance um, when you are treating them with antibiotics. As you mentioned, antibiotic regimens for TB are extremely prolonged, 6 months to 2 years. And much of the time, the patient is not showing any symptoms. Therefore, patients don't necessarily comply taking their antibiotic medication. So in many cases, it's necessary to compel patients to come to your clinic where they're given the antibiotic, where they're required to take the antibiotic. That is what is meant by directly observed treatment. In fact, directly observed treatment is practiced here on campus, New Jersey Medical School, in the ICPH building, which has a national TB center. One of its main activities is directly observed treatment of TB patients. So if you are in a situation where you suspect that you have been exposed to TB because you are a caregiver or you travel in an airplane with a TB patient or on some such situation, you will be treated prophylactically with isoniazid. Actually, I should say isoniazid and rifampicin together are given prophylactically to people who are exposed. Okay? So in our case history, the children and the husband would have been given prophylactic treatment. Now we come to BCG vaccination. BCG stands for Bacillus calmette-guerin. This is a bovine species that infects and causes a similar disease in cows, right, as the Mycobacterium tuberculosis, equivalent for cows, right? Now, BCG vaccination, which is basically an attenuated strain of Bacillus calmette-guerin that is used as a vaccine, this is given worldwide, but is not routine in the United States. Why? Well, first, for a long time, there was controversy about the effectiveness of BCG vaccination. However, the controversy is no longer as intense as it used to be. And here is our current understanding of how effective BCG vaccination is. BCG vaccination is believed to be quite effective in childhood in protecting against TB infection. However, it's not effective throughout the lifespan of a vaccinated individual. As you enter your adult years, its protective, um, um, its ability to protect you from TB infection erodes, so that BCG vaccination is not effective in adults, but is believed to be effective in children. Okay, that's the reason why it is practiced worldwide. In the United States, is not given. BCG vaccination is not given. You might wonder why. Why don't we try to protect our children from TB? Well, we don't give BCG vaccination to children because TB is a relatively rare disease in the United States, and BCG vaccination comes with a price. And the price is you lose the ability to diagnose tuberculosis because you lose the ability to use the tuberculin test in a meaningful way. If everyone is BCG vaccinated, then everybody is going to react to tuberculin test. That's the reason why we don't do that. Now, sometime in the future, when there is an alternative method, perhaps we could consider BCG vaccination, but we do not currently give BCG vaccine to children. Okay. With that, we move on to the next major mycobacterial species, Mycobacterium leprae. Mycobacterium leprae causes an ancient human disease, a dreaded human disease. What is shown on this slide on the left hand side is a victim of Mycobacterium leprae, and you might observe that this victim's limbs are both his one of his arms and one of his legs have been amputated, and his skin is shown with pockmark and deformed, and this patient is holding a bell. This is called a leper's bell. In medieval Europe, a person afflicted with the disease, when entering a public square, a public street, was required to ring a bell to alert people to his or her presence. And so this is a disease that was historically stigmatized and feared by population throughout the world. Okay? The organism that causes the disease is called Mycobacterium leprae, and there are some surprises about these organisms as you will learn. Okay, today the disease caused by Mycobacterium leprae is called Hansen's disease. That is the word preferred in the United States because of the historical stigma associated with the word leprosy. We call the disease Hansen's disease. Okay? The organism is acid fast bacillus is extremely slow growing. It's also an obligate intracellular parasite, just like Mycobacterium tuberculosis, but it cannot be grown in vitro. MTB can be grown in vitro, M. leprae cannot be grown in vitro, but it can be grown experimentally in, of all things, nine-banded armadillos and the foot pads of mice. It can be maintained in armadillos and foot pads of mice, but there's no artificial growth medium known. Very likely, and this is quite a common phenomenon for pathogens, is because of the so-called reductive genomic evolution. What reductive genomic evolution means is that a bacterium which has the full genetic capacity to survive in some ecological niche, when it becomes established in a host organism, and it becomes to then it does not need quite as many genes to carry out its life processes, because it's getting a free ride from the host cell. It's getting amino acids, and other stuff it needs to survive from the host, and therefore is going to undergo so-called reductive genomic evolution, in which simply loses many of the genes that are no longer necessary for it to live. So as a result, it reaches a point where it has enough genes to survive in its host, but it cannot grow outside that host because it requires all these other things that it needs that are not available outside that particular host. So this is quite a common phenomenon for parasites that adapt to particular hosts. So in the case of Mycobacterium leprae, no artificial medium is known where this organism can grow. The epidemiology. Mycobacterium leprae, surprisingly, ironically, is not highly infectious. So this whole precaution about people affected by this organism having to ring a bell in the public sphere because people are so afraid of catching the disease, that is fiction, as it turns out. This is not a highly infectious disease. It requires prolonged person-to-person contact. That is, typically, the affected individual is something like a close family member, like a spouse who has cohabited with the patient for years. It's not infectious like TB is infectious. It requires prolonged person-to-person contact. Mycobacterium leprae pathogenesis is somewhat similar to TB in that it also infects macrophages, but it also infects certain cells in the nervous system, like Schwann cells, and it can invade peripheral sensory nerves, which is the cause, by the way, for developing localized anesthesia, which in turn, leads to insensitivity to pain, and that in turn leads to amputation, things like that, because patients are no longer sensitive to pain. They damage themselves, and that leads to eventually loss of limbs and things like that. Okay, the immunity to Mycobacterium leprae is also cell mediated, and the disease is because of delayed type hypersensitivity response to proteins called lepromin. Lepromin is similar to tuberculin in the sense that it's also an interior protein. It's not a single protein. It's a preparation of proteins that's obtained from dead cells. Lepromin. Okay. What is the clinical presentation of Hansen disease? Hansen disease can be present in two forms: tuberculoid form and lepromatous form. Tuberculoid form is shown in the upper part of the diagrams, and its characteristic is the presence of macules. Macules are areas of the skin, flat areas of the skin, shown on this patient's face and shown on this patient's torso. Flat areas with hairless centers. In this, in this presentation, the disease is described as indolent. Indolent here means the disease is neither progressing nor receding. So there is a standoff between the organism and the host. The disease is described as indolent, and these individuals are not contagious. In these individuals, cell-mediated immunity is active. Okay. Now, tuberculoid stage is not permanent, however. It can progress to the second form, the dreaded form, the lepromatous form. Lepromatous form is shown in pictures here in the bottom right, as well as a diagnostic of skin scraping acid stain with ZL Nielsen stain, acid fast stain on the bottom right. Okay. In the lepromatous form, patients become defective for CMI. As a result of the defect, there's uncontrolled replication of the bacteria, massive growth actually, because skin scraping show massive amounts of bacteria, and the characteristic leonine or lion-like appearance with extensive skin lesions and deformation. This is the dreaded presentation of Hansen's disease. What about treatment? Here is another surprise for you, possibly. First, a few words on diagnosis. Um, Diagnosis is based on presence of acid-fast bacilli in skin scrapings and by serological tests, for example, lepromin and clinical findings. A part of diagnosing and treating Hansen's disease patients are sociological issues. Obviously, this is a highly stigmatized disease, so the healthcare provider um, has to accommodate the sensitivity of the patient to the stigma. Um, okay, the treatment of the disease is actually very simple and very cheap. A combination of dapsone, which is a sulfur drug, which is one of the earliest antibiotics in the 20th century, and rifampin. Both of them are extremely cheap drugs and is costing fractions of a penny, and they're very effective. Shown here for this patient, 6 months of treatment with dapsone plus rifampicin completely reverse nearly reverse the patient's symptoms, as you can see in this diagram. Okay. In addition to these two, a third antibiotic is added to prevent resistant forms from forming. Clofazimine is added as a third antibiotic. Okay. The bottom line is that the disease is fortunately receding from the world, and this is like definitely a disease that's coming under control. In the words of World Health Organization, Hansen disease now is considered to be a disease of the poorest of the poor. In other words, there's no reason for the disease to exist because treatment is effective and treatment is dirt cheap. Okay. Here we'll come to another surprising feature of Hansen disease, and that is the relationship between Mycobacterium leprae and a nine-banded armadillo. Okay. So it was discovered some 20 years ago, well, maybe 15 years ago. This report is from 2011. Okay. A strange phenomenon was discovered in southern United States, and that wild armadillo populations had a strain of Mycobacterium leprae. The species is the same species as what infects humans, by the way, but a particular substrain of that species was found in wild armadillo. And surprisingly, 60% of patients from southern United States had the same exact strain as found in wild armadillo populations, indicating that there was some sort of an infection process going on between the animals and the humans. Okay? And this particular strain was different from the vast majority of patients elsewhere on the planet. They had a different strain as compared to this fraction of patients in southern United States. So what's going on here? What's the relationship between armadillo and the strain that it carries and that fraction of the American patient population that had the same strain? Well, so in that sense, this disease for this fraction of the population, human population, can be considered to be a zoonosis. The word zoonosis means a bacterial or viral disease acquired from animals, okay, so wildlife. Okay, so what is the story here? Well, the story is speculative in nature. but it goes something like this. The reason that the same strain is shared among humans and armadillos is because armadillos are a reservoir of this strain, and this reservoir arose some 400 to 500 years ago during initial contact by colonizers, most likely from Spain, that is, humans who carried the disease, who somehow interacted with armadillos and seeded a population, and that organism survived in wild armadillo populations apparently of all these centuries. And the reason that same strain shows up in southern United States, in a fraction of people in southern United States, is because armadillos and humans interact in the form of, armadillos are an animal that are hunted for meat. So it's possible that consumption of armadillos or some other way of maintaining close contact with the armadillos results in transmission of the disease to the human population as well. So that so this part is speculative. What is proven is that a strain is shared between wild armadillo populations and a subset of patients in the United States. But I want to add that the disease is extremely rare in the United States, but the United States does maintain actually a Hansen's disease clinical center. It's in Baton Rouge in Louisiana. There's actually a center that treats the disease. Okay? But this disease is extremely rare in the United States. All right, so let's move on to a brief consideration of non-tuberculous mycobacteria. So these non-tuberculous mycobacteria are given various names, various abbreviations. NTM, they're also known as mycobacteria, other than tuberculosis, MOTT, also known as atypical mycobacteria. Those species are listed here. You don't need to remember all of them, but you have to remember what they are collectively. Mycobacterium avium, Mycobacterium intracellular. They go together, and that complex is called MAC, Mycobacterium avium intracellular complex. This became well known after the HIV epidemic because HIV patients were uh, often infected with MAC, Mycobacterium avium intracellular complex. Okay? There is Mycobacterium kansasii, fortuitum, ulcerans, marinum. Yeah? All of these cause granulomatous diseases. All of them are significant pathogens only in immune compromised individuals. Here is some additional information on a few important atypical mycobacteria. All atypical mycobacteria, by the way, they are environmental bacteria. They live in the soil and other environmental niches. All of them are acid fast staining. All of them cause granulomatous diseases. All of them are resistant to treatment. And as I mentioned before, they are mostly an issue only for immune compromised individual. Okay, uh, two important atypical mycobacteria are Kansasii, Mycobacterium kansasii, and Mycobacterium avium intracellular complex. Now, before the advent of modern DNA technology, these atypical mycobacteria were identified by classical methods such as growth characteristics, colony morphology, things like that. So M. kansasii is known for a specific culture property called photochromogenicity. Photochromogenicity. What this means is when you grow colonies of Mycobacterium kansasii on an agar slant and incubate the agar slant in the presence of visible light, only in the presence of visible light, the colonies turn yellow. as shown here. And that property is called photochromogenicity. So that is an identifying characteristic of Kansasii. Okay? As contrasted to the colonies of Mac, which, when grown in the presence of light, do not turn yellow, they remain colorless or white. Okay? Okay. All right. Here we come to the last part of this lecture, which is bacteria uh, that have a branch cell morphology. These bacteria are actinomyces and nocardia. Okay. In this slide, we consider a bacterial genus called actinomyces, which causes a human disease called actinomycosis. Actinomyces, actinomyces are gram-positive cells. They are, however, anaerobic bacteria. This is the only organism in this entire lecture that's anaerobic, and therefore you must know it. Okay? This is the only organism in the whole lecture that's anaerobic. Okay? The gram-positive anaerobic bacillus. It has a branch cell morphology and is not acid fast. So this organism has several distinguishing features. It is anaerobic, is not acid fast compared to other organisms, and it has branched cells. It's a feature that it shares with other bacteria, nocardia, that we're going to discuss here. Yeah. Actinomyces is is a commensal. It can be present in the environment. It can be present as a part of the normal mouth microbiome, for example. But like other components of opportunistic pathogen, under specific conditions, such as tissue damage, it can become an opportunistic pathogen. In fact, actinomycosis is an example of an opportunistic infection. Okay? A characteristic of actinomycosis is the presence of the so-called quote unquote sulfur granule. Okay? Sulfur granule is shown, a photograph is shown at the site of a lesion on the cheek of this patient. Outside, there is a small yellowish waxy substance. It's called a wax granule. In reality, it does not consist of wax. It's basically plump bacterial colonies um, that have the appearance of a sulfur granule. Okay? So what is the disease? Actinomycosis is an inflammation of the mucosal surfaces. An example of a mucosal surface is the inside lining of your mouth. That's a mucosal surface. An infection of mucosal surfaces occurs when there is damage of the mucosal surfaces. Typically, your mucosal surface is damaged during, for example, botched dental procedures, okay? Or some sort of injury that you suffer that affects the mucosal surface of your mouth, which allows uh, the um, the normal inhabitants of the mouth to invade the deeper tissues and cause an infection. Okay? In actinomycosis, the mucosal surfaces are infected, but they eventually open through a sinus through the skin to the outside. Okay? So typically, a lesion is present in the lower face and under the chin. So it's called cervicofacial location of the lesion. As we mentioned, it's associated with poor dental hygiene and botch tooth extraction and events like that. The diagnosis of actinomycosis is made on the basis of the nature of the lesion. Where is it located? Is that a sinus that opens in the opens from the inside the mouth? It's very slow progression, and a history of mucosal damage, such as a tooth infection or a botched tooth extraction, and the presence of sulfur granules at the surface, and of course, culture confirmation. Actinomycosis can be treated quite effectively with penicillin G specifically. This is the last group of bacteria we're going to consider in this lecture, and this group is nocardia. Nocardia causes a condition called nocardiosis. These organisms are also gram-positive. They're also branch, just like actinomyces, but they are aerobic, like all other organisms. All mycobacteria are aerobic, right? The only organism that's anaerobic is actinomycosis. We considered in the previous slide. Nocardia is, on the other hand, aerobic. It's also weakly acid fast. It's also an environmental bacterium found in soil and also present as a commensal. It is also an opportunistic pathogen. That is, it can coexist without causing any problem unless there is an injury or immune compromise. Okay? There are two kinds of nocardiosis caused by this organism, caused by two different strains of nocardia. The pulmonary form, caused by Nocardia osteroides, causes a bronchopneumonia with dyspnea, meaning shortness of breath, cough, and sputum, mainly in immune compromised individuals. So, Nocardia osteroides mostly infects immune compromised individuals. The other nocardiosis is the cutaneous form, and that is caused by the strain Nocardia brasiliensis. Nocardia brasiliensis typically affects feet. Okay? Shown here at the bottom right. It's also called the Madura foot. Okay? In this case, pustules and lesions with sinuses at injured or exposed sites are seen. And at one time, this kind of clinical presentation was considered to be a mycetoma. And mycetoma refers to a fungal infection. That's because people looking at the infectious agent in these cases saw branch structures. Branch structures are typical of mycelia, of fungi, rather than bacteria. So they misdiagnosed that as a fungal disease, but later it was found that they're actually bacteria, which happen to have a branch cell morphology. Anyway, uh, so Nocardia brasiliensis causes the cutaneous disease in extremities when they become susceptible as a result of injury. So typically, this is the disease that is caused occasionally in people doing manual labor, farmers who expose their feet to injury while being exposed to the soil. Right? Diagnosis is based on basis of presence of gram-positive branch cells in sputum in the case of osteroides, or in aspirates in the case of the cutaneous form of nocardiosis. Treatment is quite effective. Sulfonamides are quite effective in treating the disease. So we come to the last slide where I have prepared a summary table to highlight important features and differences between the three different sets of bacteria that we considered in this lecture. On the left column are the three major groups: mycobacteria, that includes everything: TB, leprae, and, and atypical mycobacteria. Then actinomyces, and then nocardia. The gram staining status is shown here. All of them are gram positive. Okay? Specific staining feature, acid fast staining feature. Mycobacteria are all acid fast. Actinomyces is not acid fast. Nocardia is acid fast, but weakly so, but still acid fast. Growth characteristic. Mycobacteria are always aerobic. Actinomyces is anaerobic. Nocardia is aerobic. Cell morphology. Mycobacteria, all of them, are slender rods. Actinomyces, branch cells. Nocardia, branch cell. Okay, that concludes our lecture. If you have any questions, I'll be happy to answer them. Otherwise, best of luck in your exam coming up.
Feel free to mute if you have any questions you'd like to ask, or unmute.
Yes, please unmute yourself. I have a chat question here now. No.
Okay, it looks like we're good, Zafri. Don't forget to stop your recording.
Yeah, good idea. Oh, dear God. Bad news. I
You're still recording.
am? Okay. All right.
Yep. Yeah, I can see you. You're still recording. So just stop it.
All right.
There you go.
I
You're
thought
good.
I forgot to restart it.
Okay.
All right. Very good. Okay. Yeah,
All
we're
right.
back.
Professor, I just have one question. It
Yes.
might be in your PowerPoint notes, but I would like clarification. With reactivation of TB, is that different from latent TB, or are those both the same?
Okay. Latent TB is not an active disease at all. Okay.
Right.
Reactivation TB happens among people who have latent TB. A small subset of them suffer reactivation of the tubercles.
Okay.
So,
Okay.
so reactivation TB can only occur in that population which has latent TB.
Okay. All
Right?
right. Thank you. That makes sense. Thanks.
Okay.
All right. Thank you, everyone. Thank you, Zafri.
Thank you. Bye-bye.